So why don't we get started? Just a small announcement before presenting our speaker. Stick around after the talk today because there will be pizzas. Stay around to chat about science and eat some pizza. And it's a pleasure to introduce Avihu today. Avihu did his PhD here at the Hebrew University. Avihu did his PhD here at the Hebrew University, followed by a postdoc at Columbia University with Professor. And he's going to talk today about two recent projects published in eLife and Science of Dances, looking at motor circuitry in birds and chicks. So please come. It's great to have you here today.
Thank you, Aya. First time ever being here, so pleasure to be here. Okay, so I'll tell you the study that I've done in my labs in the past few years. Maybe I'll start with this introductory slide, focusing on spinal cord circuit to control various modes of movement, locomotion, flying, and so forth. So if you look here, this is a human being. I mean, some people think he's not a human being, but I guess genetically it is. And the birds, so they are running, so they are using their legs in an alternate fashion, which is something we all do, not only bipedal animal like birds and human beings, also quadrupedal animals like horse and mice, of course. They walk via alternation of the leg, and this activity, although it's controlled by our brain, but actually the spinal cord is acting autonomously in order to activate this kind of movement. Here you see other modes of movement where the legs or wings are moving together in synchrony, so kangaroo, birds, or bats, and the question whether these kind of activities are also modulated by hardwired circuitry within the spinal cord, something I'm going to discuss in the second part of my talk. And over here you see another human being, which is the question whether human being is still valid. This is Pelé scoring a goal. So this kind of movement requires not only automatic behavior of the spinal cord circuitry, but also requires a constant feedback between the legs and the brain. So the brains need to know what the legs are doing. So in the first part of the talk, I will discuss how do we know what our legs are doing. But before I start, I would like to mention the one that did everything. So this is Baruch Haimson. This is in his PhD period. You can see he was very neat, elegant, and he used to smile a lot. And this is during his postdoc over here. I'm a bit concerned, I must say. And it is, what is saying here? I want to be free. Okay. Now you want to be free, come back to the Hebrew University at Boston Medical School. So this was mainly his project. And the first part of the project, which I'll describe in the first 20 minutes or so, is a collaboration between my lab and the lab of Professor Aharoni Lefter in our department. So let's start. So we see here making Refinement want to score a goal in the final Mondial for female USA against the Netherlands. In order to perform this amazing task, first of all, she needs to want to kick the ball. So there is the cortex is planning what's going to happen soon, and then the command is being relayed to the spinal cord, and here motorneuron eventually execute the action of the leg. But this is not sufficient because there's a long way between here and the ball, and during this movement, the brain should know what the leg is doing in order to succeed in scoring the goal, as it actually happened. So while the leg is moving, there are two kinds of feedbacks which are being relayed to the cerebellum. One is actually what happened. So this is a sensory modulation, mostly the activity of the muscles, what's called proprioception. The state of the muscle and the joint is being transmitted to the cerebellum, and it is compared with another input, actually is a copy of the motor command. So it was postulated that once a command is being relayed from the spinal cord to the muscle, a copy of this command is being relayed also to the cerebellum. So cerebellum gets simultaneously the results and the actual action. If there are discrepancy between them, there's enough time to fix it and eventually be able to move the leg toward the ball in a successful manner. But it is known about this pathway. Other indeed neural neuron which project to the cerebellum? And the answer is obviously yes, but how do they actually are required for accurate stepping or kicking the ball? So this is something that is not known. So this is just a scheme. So the intention is being born with a cortex, related to the spinal cord, and then to the leg, to the musculature, muscles in the leg, and then the proprioception, the sensory information is relayed to the cerebellum together with this pathway which is called the VSCP, the ventral spinal cerebellar tract, which is actually a copy of the motor activity which is being read simultaneously with the sensory information. And then the cerebellum and the cortex update expectation, and if it's required, there is a tiny changes in the way the leg is moving, eventually to lead to the proper kicking of the ball. Okay, so studies done mostly in cat by this group of scientists, many of them, most of them in Sweden, have identified in a physiological way and anatomical way a group of neurons in the lumbar spinal cord. These are the region which control the musculature of the legs. And this neuron, they propose, are the one which send this copy of motor activity to the cerebellum. And they, using a various old-fashioned technique, but still a very good one, they were able to map the trajectory of this neuron. So this neuron here in green project to the other side of the spinal cord, what we call our commission neurons. And then they project all the way to the cerebellum, and in the cerebellum they cross back to the other side. The excitatory neuron, they're also inhibited by premotor neuron. So this red neuron over here is the premotor neuron which activates the motor neuron. It also sends axon which innervates this VSCP neuron. So the VSCP neuron actually gets the same command just like the motor. So this is basically the copy of the motor activity with this neuron, which project all the way to the cerebellum. We also saw that this neuron projects to the other side of the spinal cord within the lumbar level. They receive sensory information, here in yellow, as well as descending pathway from the brain. So this list, checklist which I'll go on through my talk, is a finding that was done mainly in cat. And they propose, according to the wiring of this neuron and by recording their activity, that this neuron sent a copy of motor activity to the cerebellum. However, the actual role of this neuron in stable locomotion was not challenged because there was no way to alter the activity of this neuron because there was no genetic means to target this specific neuron. So the project we took a few years ago, together with Barb, of course, was to find a way to genetically enable this neuron to follow the projection, to see whether they fulfill all these checklists, all these things that have been proposed by studying the cat, and see whether they are indeed required for stable locomotion. So I'll start with a short clip. So here, this is a cross-section of the spinal cord, early stage of development. We are focused on 3 types of dorsal neuron. The focus will be in VA2. So these are the second dorsal neuron which are born at the dorsal spinal cord. This is the time scale of development, and we're dealing with a chick embryo. So this will go on. So let's have a look. Okay. It's running. Yes, it's running. Okay, so there, this VA2 neuron project to the floor plate, the ventral midline, cross to the other side, and then they project rostrally towards the brain. So here this the sense collateral, this is at the lumbar level, the sense collateral, but this collateral also spread along the entire axis all the way to the brain. And this is already E17. This VA2 neuron project to the cerebellum. So this is, I'll show you all the data soon. The project to the cerebellum, cross to the other side, just like the VSCP neuron. These are innervated by premotor neurons, here in yellow and red. They send collateral to the other side, which innervate a premotor neuron at the other side, as well as the lumbar level, as well as the entire axis towards the brain. So this is just a short clip, but I'll show you the data now. Okay, so I'll go through this checklist and I'll show you that what we have found fulfill most of the criteria laid down by It's the people that study the VCT neurons. Okay, so this is the DI2 neurons here labeled with GFP. So we're using the chick embryo as a model organism. It's very easy to manipulate these cells genetically within the embryo. And it's they're also bipedal, which is by itself extraordinary. So DI2 neuron, this is the cell body. You see the axon projected to the other side. So they are indeed commercial neuron. They were able to target DI2 by using a combination of enhancer element, which I won't discuss this in detail this time. There are a excitatory interneuron. In order to reveal this, what Baruch did was to label DI2 neuron with GFP, and on top of it to perform in situ visualization with BGLU2, which is a marker for excitatory glutamatergic neurons, and about 85% of DI2 neurons at the lumbar level are excitatory neurons. They're connected to cerebellum, so we label, so the advantage of the chip that we can actually target labeling to a very defined level in the rostral level. So we can deliver the GFP only at the lumbar level and then follow the projection of the axon all the way to the cerebellum, unlike the mice where the labeling is genetically. So we see everywhere here we can, by using a topographic means, we can actually label just a portion of neuron within the lumbar spinal. So this is the lead over here. So we label the neuron at the spinal, at the lumbar level, and then follow projection up to the cerebellum. And you can see here, this is the DI2 neuron, which are born at the lumbar level. They enter the cerebellum. זה לא יפעיל את הכיסא מפלט? יותר טוב? נמשיך ללחוץ? אוקיי. Okay, so here shows. Okay, so you see this is a DI2 axon entering the cerebellum and cross to the other side, just like it was shown in the cat for the VCT neuron. So they projected the cerebellum. Another way to show it, I saw you a short clip. So Baruch did a light sheet microscopy of an entire spinal cord. So we labeled just the lumbar neuron at the lumbar level and then follow the projection along the entire axis using light sheet microscopy. And this clip is so this is the red in cerebellum, the blue is the brainstem. So these are the DI2 neuron entering through the brainstem. Here, entering the cerebellum. And crossing to the other side. These are the actual traces you can see here. That using these traces, Baruch was able to construct these images. So these two neurons, there are many others that we label in this video clip. They actually, the cell body are within the lumbar spinal cord that project all the way to the cerebellum, and just like the VCT neuron in the cat, they cross to the other side. So they fulfill this criteria as well. They are also innervated by premotor neurons, so we devised a way to label not only genetically, not only DI2, but also premotor neurons. So this red neuron over here is an inhibitory premotor neuron, which we have means to label it genetically, and we express synaptic sharing in this red neuron, and in green, this is the DI2 neuron. You can see there are multiple boutons on top of DI2 neuron. So DI2 neuron, this supposedly VCT neuron, receives synaptic input from inhibitory premotor neurons, and we did something about the same with excitatory premotor neuron and got the same result. So DI2 received premotor neuron information, inhibitory as well as excitatory. DI2 neuron also sends collateral. Here you can see this image. You see the spinal cord labeled at the lumbar level over here. And you see the axon of joint longitudinally toward the cerebellum. That sends all this collateral inside. You can see it here. as well as in this cross section. And now you see it in a short video clip. So this is a lumbar spinal cord. I refer to this structure in my second part of my talk. So this is a bird-specific element, which is only the lumbar spinal cord that I refer to later. So this is just the lumbar spinal cord, now stripped from the, now you see the neuron and the axons. This is midline, cells are over here, axon growing over here. You can see these are level 2 neuron, and you see all these collateral which originate from this DI2 neuron. The main axon goes all the way to the cerebellum, but it sends collateral all the way through at the lumbar level, as well as very soon also in more rostral level, even in the wing level.
Question.
Sure.
You mentioned inhibitor and excitatory input.
Yes.
Are they spatially organized?
The synapses themselves.
Yes.
In both cases, we saw the synapses on mostly on the dendrites. We had
Excitatory.
also,
You showed
also in the soma, but they were also in the dendrite. The labeling system is a bit tricky, so it's easier to see the cell body, the soma, and sometimes the dendrite it's hard to show. But we saw dendrite buttons on both soma and the dendrites. And we didn't look for any specific organizations, so I don't know. Okay, so using all this genetic trick, we were able to actually map what are the targets of DI2. So this is DI2 neuron, which projects all the way to the cerebellum and sends collateral. It's innervates DI2 from the other side. It innervates premotor neuron. These are ipsilateral premotor neuron, but also innervate commercial premotor neuron, and it also innervates premotor neuron at the wing level. So this is a very complex pattern of innervation. It sends the main axon to the cerebellum. However, it sends many collateral all the way through at the lumbar level, thoracic level, wing level, brainstem, and so forth. So one neuron is able to innervate multiple targets, and we can discuss later whether this means that this report about motor activity is being sent only to cerebellum, but also maybe all being sent to local target within the spinal cord. So this is something it's indeed an option. So DI2 innervate collaterals, sends collaterals innervate spinal motor neuron along the entire axis. It also receives sensory input, so here we label a sensory neuron using a specific reporter element. And you can see here, this is in green DI2 neuron, in yellow, these are synapses which originate from the sensory neurons. It also receives input from sensory neurons. The only thing which is lacking in our checklist, it doesn't receive input from reticulospinal tract neurons. So these are 5HT neurons, and since the 5HT synapses is the lumbar level, doesn't touch the DI2 neuron. Probably there are other VST neurons, DI2 not the only one. So they fulfill many of the criteria, but not all of them. Okay. So in order to challenge actually the role in locomotion, we set to target this neuron genetically, and we express using our genetic system tetanus toxin only at the lumbar DI2 neuron. So this is being done. So this is a scheme of the experiment. We introduce the tetanus toxin at early stage of development into the embryo only at the lumbar level. Then we let them develop in the egg. They hatch. In the first 3 days, we need to imprint them. So Barbara was there and calling them name and sing songs for them, or to imprint them, because eventually the task will be to run toward the trainer. And this at day 8, we actually let them go, let them run, while we film them in a high-speed video camera, actually iPhone. And then we extracted the data out. Okay, so we had, so this is a very challenging system for actually getting able to hatch after genetic manipulation is quite unique by itself. Many of them failed to hatch and died, but the few that did were okay in terms of strength. Instead of size, the size was equal to the size of normal hatchling. In terms of strength, the grip strength was similar to the control hatchling. They also worked in terms of alternation between the legs. They worked normally, so the phase between the legs was okay. So they can walk, they can hold, they can tweak, they can eat. And then we tested other features of the locomotion. What you see here, this is a series of several steps. This is a control embryo, just expressing GFP. And this is the knee, the ankle, and this is the TMP. Birds have an extra joint called the TMP joint over here. And basically, when birds are walking, they don't really, I mean, the main elevation is being made by the ankle joint, unlike us, where the elevation is by the knee. The bird is mostly the ankle. Okay? The hip is quite stable. And this is a profile here. This is a profile of the knee in view of the ankle and so forth. So this is a normal stepping pattern. There are variations between each step, as you can see here. And this is a series of few steps of genetically manipulated embryo. And you can see right away that the steps are much more different. So this is over here is a kind of, there is a fall, collapse, and here there is a upshoot of the leg. And overall, it seems that there is a lot of variation in the profile of walking, which quite reminiscent for us of maybe how ataxia syndrome is how it looks in human beings. So we basically measured from each hatchling between 50 to 150 steps. This is just putting all the profile together. So this is for control, just looking at this profile, but putting all the profile now together. So this is in control embryo. See the extent of shomut or Halimila. Is very small. However, here in the manipulated, the extent of variation is the range is much higher, as you can see here. When we plotted the range, you see a very robust differences between the control and the manipulated embryo. This is just looking at the height of the knee. We did a similar experiment looking now at the P and P angle. We see similar phenomena. So this is the control, and here the extent of variation, the variability is much higher in the manipulated embryo, as you can see here also in the quantification. And so we saw that embryo hatchling which were genetically manipulated, the activity of gait2 was inhibited by expression of the tokenist taxon. They walked in an unstable manner. So just to summarize this part of the talk, so gait2 neuron, the wiring pattern is very similar to the VACT neuron. They project to the locomotion neuron, excitatory neuron, they project to the cerebellum. They receive a premotor neuron information at the lumbar level. They also innervate premotor neuron at the other side of the lumbar, at the contralateral side of the spinal cord, at the lumbar level, as well as in the wing level. So the question whether what we've seen just now, the instability in locomotion, relies on the projection to the cerebellum, or maybe the local, maybe gait2 neuron actually are reporting to the other side what this side is doing require for stable locomotion. These are still open questions. It's very hard to challenge them because the complex wiring is the same neuron projecting to the cerebellum as well into the other side of the spinal cord. So it's very hard to genetically manipulate only this input rather than the cerebellum input. So we are still trying to figure out how we can actually challenge this idea. But I think this result was the first time we were able to label this presumably neuron that sends a copy of motor activity to the cerebellum and shows that they are required for stable locomotion. Okay, so from stable locomotion, I move to hand waving. And this is not a good organism for hand waving because when we walk, we move our hand via alternation. So birds are much better for this. So they have this very unique phenomena. They fly by moving their wings together. However, they walk by using the legs via alternation. And it was shown already by using classical transplantation experiment. The spinal cord of the, at the brachial level, at the wing level, is sufficient to instruct wing flight. So if you take this region, put it here at the lumbar level, the leg will move in synchrony. So all the information is required for synchronous movement of the wings is within the spinal cord. And vice versa, if you take a spinal cord for the leg, put it here in the wing will move in alternation. So everything which is required for either flying or walking resides within the specific level of the spinal cord. This is how mouse is walking via alternation. And study done studying at the turn of the 20th century, like Thomas Graham Brown, and later on by Stan Jr. and this group of scientists using mouse as a motor organism, we actually mapped the circuitry which is required for alternation. Okay, so what you see here is a very schematic wiring pattern of the spinal cord with instruct walking alternation. So this green neuron represents many different types of neurons, but they are all excitatory interneurons, which activate motorneuron at the same side, whereas the blue neurons are inhibitory neurons, which innervate motorneuron at the other side. So co-activation of these interneurons, the premotor neuron in green and the premotor neuron in red, will activate this limb and inhibit the other limb. So this idea was laid down already by Thomas Graham about 100 years ago, and it was shown genetically in mouse in the last 20 years. So, and of course, this pattern of wiring, which is required for alternation, is instructed by the way the neuron projects their axon. So the blue neuron should cross to the other side, and the green neuron should stay at the same side. So the way the axon are growing should be controlled in a very precise manner. So eventually this will need to go and stop. And this molecule over here, this is a process with the spinal cord, and this is a guidance molecule which is called F3B3. Okay, you see here. And this molecule serves as a stop signal for the green neuron. So green neuron won't grow to the other side since this molecule is preventing them from crossing. So this is an inhibitory guidance molecule that prevents only the green neuron from crossing to the other side. So this is FMB3, and this is its receptor, FA4, okay? And the lack of this molecule would lead to this hopping phenotype. Okay, so this mouse is lacking the FMB3. In this case, this is a receptor, but the phenotype is the same. You should see this movie again. He's hopping. He can't walk via alternation just because this molecule here is absent, and this absence of this molecule will lead to now to excitatory neuron crossing to the other side and innervating motor neuron at the other side, and eventually we have a simultaneous activation of motor neuron on both sides. So this was known in mice using genetic mutants and so forth. And so knockout of FMB3, its receptor FA4, or a signaling molecule which is downstream alpha-2 chimerin, they all lead to a hopping. So this is in mice. What about birds? So we thought maybe this is what happened in birds. Maybe birds, so this is the legs of a mouse, normal mouse, this is the legs of fusion mouse. Maybe this is what happened in the legs of chicken or bird. This is what happens in the wing of bird. Maybe an absence of a gene only at the brachial level would actually instruct the core flapping in the wing versus the walking of the leg. This is an idea. And we, together with Claudio Mello from Portland, we set to look for the FMB3 gene in birds. So starting, this is a whole animal kingdom almost. So in red, this is FMB3, this molecule which prevents axon from crossing. Okay. This is its syntenic structure in the chromosome. It's always around this blue gene, which is around this yellow gene, and so forth. You see, this is conserved from frog, human, mouse, crocodile. Birds lack this blue gene. Chicken lack FMB3 gene entirely. Chicken, quail, and the whole family of chicken-like, which is called galliform, they lack the gene completely. Other birds, mostly songbirds, do have the gene. However, the protein structure is a bit different. So this is the structure of FMB3 gene in mammalian and reptilian, quite similar. However, birds are lacking several modules. So we had to check whether the bird's protein is functionally different, and we actually wanted to see whether the protein, the FMB3 protein, is able to bind its receptor. So remember, in order to prevent axon from crossing, the neurons are expressing a receptor, which is called F4 here. The receptor will bind to this guidance molecule, and once it reaches the guidance molecule, it will repair and stay at the same side and then cross to the other side. This is just a simple assay. When we express the mouse protein or the crocodile, alligator protein, and use this as a bait for receptor, which is fused to a color alkaline phosphatase, you see that we get a nice color, meaning that a receptor will bind either the crocodile or the mouse protein, but it won't bind the zebrafinch. So this is a bird, this is our model organism, zebrafinch. It doesn't bind the zebrafinch protein, either in this one or no, yes or no assay, or we need a more quantitative assay. The bird's protein doesn't bind the receptor. So it seems that in birds, several events have happened. Some of birds lost completely the gene. Some of them, the protein is altered, and we also showed, which I won't show you here, that the regulation of expression is different in birds than in reptiles or mammalian. And eventually, the protein is not active in birds. So that this would actually lead to the wiring. So this is how, this is the mouse, let's say, this is the mutant mouse. Does the wiring in the wing level of the bird looks like the mutant mouse? So in other words, does bird learn something from mouse genetics and acquire these tricks of knockout to be able to fly? So whether a naturally occurring knockout of the FMB3 resulted in wiring which instruct flight. So you're already acquainted with the checking system that I have. So just looking at the mouse, so this will be wild-type mouse, this is a mutant mouse. There are several features which are different between these two spinal cord. First of all, the roof plate here, the dorsal part of the spinal cord is small in the mice. It's very large in the mutant mice. There are ectopic crossings, so the axon will cross at the dorsal midline. And there is more excitatory interneuron now which innervate motorneuron. So we said to test all these features. So this is again wild-type mouse, the mutant mouse. This is the central canal here. This is the ventral midline, dorsal. Yellow is big, red is small. Mutant, red is big. And as well as in bird. In bird, the dorsal midline is always bigger. What about crossing? Let's see. This is the leg level of chicken. This is the wing level. You see here is the wing level, axon cross here, below the central canal, and also above the central canal, which you don't see in mice. And this is the leg level of chick, so there is a difference in the way that the axons are wired in the wings or in the leg. Here, they cross only under the central canal. Okay, so this is the spinal cord of the chicken, and this egg is not an egg. This is a bird-specific organ. Only birds have this organ. It is stuck within the spinal cord, just above the sciatic plexus, with several dorsal spinal cord. You see it here in the cross section, and this we assume that this structure co-developed in chick together with the loss of activity of this guidance molecule in order to actually prevent axon from crossing and And the lack of FMB3 is being compensated only in the leg level with this bird-specific organ. There are some evidence that dinosaurs also had this organ, but if you reptiles, they don't have it, mammals don't have it. Nowadays, only birds, which are the modern dinosaurs of our era, they have this specific structure. So we think this structure evolved in birds in order to enable them to walk versus the wings to fly. What about the way that axons are crossing? So this is a leg level. We use a method which is quite similar to rabies viruses in mice to label premotor neurons by injecting the virus to the musculature of either the legs or the wing. So here we use this method to label the premotor neuron of the leg. You see they're all under the central canal. They all cross here under the central canal. However, here in the wing level, they cross also above, which is quite reminiscent for what we see in mice. Over here, so in birds, axon cross also in the dorsal midline, similar to what we see in muted mice. And also Bauer tests how many premotor neurons are either hereditary or inhibitory. In this new group of interneurons, which is only in the wing level, the number of hereditary neurons is higher than at the leg level. So there are more hereditary interneurons which cross to the other side at the wing level. Okay. So it seems that the wiring at the wing level of the cheeks of the birds, when we looked at the chick as a zebrafinch, is similar to the muted mice. So the question is whether we can bring back from the future FMB3, use the mouse gene, and put it back inside the bird spinal cord, and somehow to reverse the wiring from the wiring which support wing flipping to the wiring which support walking. So what we try now is to express here in the roof plate. So let's this is just a schematic of the system, but eventually we are able to express here as a dorsal midline the mouth gene FMB3, which is lacking in bird, and see how it may affect the wiring of the spinal cord. So here, this is just the control. Here in magenta, this is where the GFP is expressed. You can see axon crossing the GFP area. However, when we express FMB3 in birds, now axon fail to cross this region. So it's sufficient to express this protein and to prevent axon from crossing to the other side. And also the premotor neuron, now Bauer combined the two methods, so labeling the brachial spinal motor neuron by injecting the virus into the wing musculature, mostly to the pectoralis, the chicken pocket muscle, and then together with the expression of FMB3 over here. So this is a control, axon crossing here with the dorsal midline above the central canal, but once we express FMB3, here in red, they fail to cross. So it's sufficient to express FMB3, and now premotor neurons, which are usually cross to the other side, they don't cross. And since the select operation is a transient method to express gene, and there is the efficiency varies between each embryo, you can see that there is a very nice correlation between the efficiency of the expression and the phenotype. The more expression you have, the phenotype is higher. You see here, this is the wild type, many axons at the dorsal midline, and when you express FMB3 high efficiency, it's all neuron drops down, now they cross here under the central canal. So we were able to actually manipulate the wiring, and now within the wing level of the birds, axon looks like a wiring of stepping mice. So the question is, do they fly? It's a good question, we don't know yet. So here we have a basic problem with the system itself. It seems that in order to achieve this high efficiency, you eventually kill the embryo. So this is something we look at within the egg. So E17 is comparable to P3 mouse, but this embryo failed to hatch. I guess it was too aggressive for them. This whole system of providing DNA inside the embryo using electroporation. Now we are trying other methods. So it's still an open question whether it's sufficient to express this gene from mice in order to change the behavior of the way the birds move their wings instead of synchronous flipping to alternation. So this is still an open question, and we're trying now to use new genetic means in order to provide this gene and see how it may affect the way the birds are flying or not flying. Okay, so I'll show you in this part of my presentation that it's a wiring within the spinal cord of birds looks like the wild-type mouse, but we don't have the FMB3. Instead, we have this glycogen body, this very unique organ, which is a bird-specific organ. And it will, however, the brachial, the spinal cord in the brachial level looks like the mutant mouse. And we were able to shift the wiring when we provide the gene back, express FMB3. The wiring in the spinal cord now resembles that of the wild type. And still, the bottom of proof whether this is sufficient to elicit walking on the hand is still something that we need to do. Okay, so I'll stop here and just mention. So as I said, most of the work was done by Baruch. Gerard did a lot of biochemistry, which mainly dealt with the ways that FMB3 of the birds bind its receptor. The Ut did the study which I didn't describe here about the regulation of expression. And this was the first part is collaboration with Aaron Lepto, Claudio from Oregon Health Science University helped out with all the genomic information. The walking of the chip was done in the Kami Center with the help of Yuval Cinnamon. That's it.
Very nice is the different genes expressed only during development, or this repression is required throughout the life.
Okay, so there are 3 FM genes in
Yeah.
the mice, 1, 2, and 3. So local FMB3 is sufficient to only affect the mode of locomotion. So I guess B1 and B2 compensate for the loss of FMB3. A lot of studies is required for synaptogenesis and so forth. However, in vivo, the lack of FMB3 leads mainly to hopping phenotype. Maybe they are less intelligent.
What is the importance of the cross inhibition? Told us about the cross excitation.
Yes.
What about cross inhibition?
Okay, so the same. So if I just go back one slide, I won't show you the entire thing. Yeah. Okay. So cross inhibition. Okay, so there are guidance molecules which are required for attracting this neurons. So NETP1 is also NETP1 is an attracting molecule, which actually causes this crossing, and the lack of NETP1 leads to similar phenotypes. The mice they don't live, but they theoretically they hop. So if you take a spinal cord of a mice which lack this attraction molecule, the pattern is.
So you need the cross inhibition to provide better walking.
So you need both of them. So I mean, but
I mean,
it's enough.
asynchronous.
It's either you are, if you like this, allow this projection, then you hop. You prevent this one, then you hop also. Then it's very, I mean, there are also extended neurons which cross to the other side, but the extent of cross excitation is lower than the extent of cross inhibition. So this is very, very simplistic. So there are also extended neurons which cross to the other side. So when you decide to hop, you can do it. Yeah? You can do it because maybe your brain inhibits this neuron and activate cross excitation. So cross excitation is there, but to a lower extent.
And what is the organ of the cerebellum? Because the work from Megan Kelly, she saw that if the cerebellum.
Okay, so we don't. I mean, the wiring. So this is my first part of my presentation. So this group of neurons which project to the cerebellum, they also project to the other side of the spinal cord along the entire length. So we can't actually differentiate between its role in reporting to cerebellum or reporting to other sites. Theoretically, we would like to assume that this has to do with related to cerebellum, but as to now, we are not sure about it. Maybe it's sufficient just to report to the other side, and this eventually will lead to a stable locomotion. Nobody knows.
Thank you very much, Professor. You mentioned that this organ, its function is mainly to prevent the crossing.
Maybe you can ask without the mask.
Yeah, sorry.
Yeah.
You mentioned that this organ, its main function is to prevent the excitatory
It's
or
what we
one
would
of
like
its
to
main
do.
functions. So that means that in evolutionary perspective, the chicks were hopping before walking? Because if you say that they evolved this organ for preventing the crossing, then they were crossing before.
Well, I don't know. But it's, first of all, it's originated in dinosaurs. Okay, so we would like to believe that it's co-evolved. So I don't know whether what happened prior to what. So dinosaur were the first bipedal animal actually. So whether the ability of dinosaur to walk on two legs has to do with development of this organ, it's an open question. I need the genome of a dinosaur to resolve it. I don't know. I mean, I would assume there is co-evolution. What came first? I don't know.
You mentioned also that where the organ is, there's also some crossing in the bottom part of the right. That's not enough to cause excitation of the other side.
It's a good question. That's but it's a good question. I asked this neuron, what kind of neuron are crossing here?
Excitatory ones that are crossing.
So according to our results, most of the axons which cross the outside are in vitro. Okay, about 60–70% are in vitro.
For stability, I would guess you need to know what the other side is doing at the same moment, and the other side is doing a moment before, which may involve cerebellum. Not just the variability, but to what extent is a different pattern in a step, and what you'd expect compared to what I guess the position of the other leg versus the position of the other leg previously. With conflict with that would help. You could see which pattern you expect for a given leg in the jigs, the impact jigs, based on the pattern in the second leg, the current position, versus the position in the previous leg.
Vision, how would the next step would look by looking at the previous step? Something like this.
Guessing, no, I mean, one leg further than another, that's part of the disease. That step to show if it may involve also something longer than just immediate projections.
Sure.
How does the circuit look in rabbits compared to mice?
Okay. So rabbits, the question whether rabbits are naturally hopper or naturally walker, it's a good question. So I mean, even mice, when they accelerate the pace from walking to running, they change from walking to hopping. Rabbits, when you just put in the ground without the wool for human being ground, they will walk, won't hop. And the wild looks like in the mouth. So the question is, what prompts the early hopping of rabbit versus the one of mice or antelope? This is probably this descending pathway from the midbrain. This is a center which projects a transition from walking to galloping to hopping. So I guess in rabbit, this center is strong. This happens quite earlier. If you take just a spinal cord for rabbit and you record activity in the dish, you'll have alternation, just like in mice.
Collaterals of the EI2 interneurons. Do they have a pattern? I mean, they say the
collaterals are sent every other segment, or they are just random?
Quite randomly. So when you see this short clip, we didn't see a pattern of entering every 2 micro. We see it kind of seems like quite random. They enter at multiple levels. We haven't seen any pattern of entering the spinal cord. They do avoid motorneuron, so we always circumvent motorneuron either above or laterally. They won't touch motorneuron, and they just sense collateral. So we haven't seen any very definitive pattern in the way they do.
It would be bizarre because you would expect to have all these attractive molecules to attract collaterals or repulsive molecules to repel collaterals in quite a patterned way. Okay. Question number 2. Remind me, when are the contralateral projections taking place vis-a-vis the development of the glycogen body?
Before.
Glycogen body appears by day 7.
Yeah, no, before. So this is like 8, 9, and this collateral is kind of 1 day before. But the entities are over here. Nothing to do with the glycogen bodies. They don't touch it.
No, I'm talking about the dorsal.
Ah, the dorsal. Okay.
The dorsal.
They start quite late. At 8, 9,
Because
10.
otherwise you cannot
It's
propose
late,
that
yeah.
the glycogen body is
Yeah.
alone.
It happens later in development. This is why most people miss it. Dorsal crossing. Never happens because nobody looks at later stage of development.
Great. Thank you
Yeah.
very much.